Hello, my name is Dr. Paula Smith-Brown. I'm a PhD qualified accredited practicing dietitian and the clinical science lead at Microba. In this tutorial, I'm going to give you a brief overview of the mucin degradation result in the Insight report to help you utilize this important finding in your clinical practice. Microbiomes are able to use fiber, protein, and mucin as a fuel source. In the Dig Deeper section of the Insight report, you'll find a tab called Microbiome Digestion Potential. In here is provided an estimation of the microbiome's potential to utilize fiber, protein, and mucin as a fuel source. Mucins are molecules known as glycoproteins that together form the mucus layer which lines and protects the gastrointestinal tract. By glycoprotein, we mean that it's both a protein and a glycan. At its base, it has a protein backbone, which is elongated by oligosaccharide chains known as glycans. These glycans make up about 80% or over 80% of the weight of the mucin molecules and are highly diverse. 56 different glycan structures have been reported in the literature, but these are all just different variations and combinations of 5 monosaccharide building blocks. These mucin molecules then cross-link with each other, which together form a thick, viscous mucus layer within the gastrointestinal tract. The colon mucus is characterized by a dense inner layer, which is impenetrable to bacteria and provides some separation between the microbiome and the gut epithelium, and a looser outer layer, which provides a 3D structured environment for the microbiome to live within. Microbes are able to use the mucin glycans as a fuel source. Some bacteria break down the glycan chains into the monosaccharide building blocks, while others burn the monosaccharides for fuel. The mucin degradation results within the Insight report gives an estimation of the number of complete functional pathways for breaking down these glycan structures. Some mucin degradation is important to maintain turnover of the mucus layer within the gastrointestinal tract. In fact, it's been shown that in patients with ulcerative colitis, their microbiomes actually have a reduced potential to degrade mucin, which may be impacting their capacity to generate the beneficial short-chain fatty acid butyrate. In contrast, excessive mucin degradation can disrupt the mucus layer. For example, in patients with diabetes mellitus, it's been shown that there is encroachment of the microbiome into the mucus layer. And because the mucus layer is not as tightly packed, this means the microbes can move deeper within it and get closer to the gut epithelial layer. In one study, it was shown that the distance between the gut epithelial layer and the microbiome was 3-fold shorter in patients with diabetes compared to healthy controls. Most species of bacteria that can use mucin can also use fiber and will only turn to degrading mucin if sufficient fiber is not available. Human studies have confirmed that high fiber diets are associated with a reduction in mucin degradation in the microbiome and, reassuringly, an increase in the generation of beneficial short-chain fatty acids. In summary, the mucin degradation result within the Insight report completes a picture of which fuel sources the microbiome is geared towards consuming. Excessive mucin degradation can be a risk factor for disruption of the mucus layer that lines and protects the gastrointestinal tract. And it can be an indication that the diet does not contain sufficient amounts of slowly digested fiber. I hope you found this tutorial helpful. If you're interested in learning about the other recent report updates we've had, please do visit the other tutorials, which can be found within the video tutorial page of the Clinical Resources Hub. If you have any questions or queries, please contact us at healthcare@microba.com. Thank you so much for your time.